welcome back, and let us welcome back to this immunology lectures, and we will be talking today about the effector T cells. So, for the last 4 classes we have been discussing about the maturation and activation of the B cells, that is the humoral branch of the immunity. In one of my beginning lectures, in the starting lectures, I had already discussed a little bit about the adaptive branch of the immunity. So, the adaptive branch of the immunity comprises of signals that are processed from the innate cells, which are primarily the antigen processing cells, and they are being processed and presented. So, these antigens are presented to the T or the B cells, and the adaptive immunity clearly has 2 branches. One is the humoral branch, which is the B cell mediated and antibody mediated branch, and the cell mediated branch, which is the T cell mediated or the T lymphocyte mediated branch. So, these antigens are usually carried by the dendritic cells or the macrophages, which present it to the T cells, that is the naive cells, the naive T cells or the naive B cells, and then activates them, that activates the adaptive part of the immunity. Now, from the earlier lectures you have learned quite a little bit about the T cell development, the B cell development, the T cell development, antibody mediated effector mechanisms, the responses that are generated by the antibodies. We will learn more about the antibody responses. We have not yet discussed about the complements, the complement proteins, the complement systems, that we will be discussing very soon, which are the antibody mediated effector mechanisms. So, we have kind of learned some of the effector mechanisms by the T and the B cells, and also the development of these 2 cell types, like the T cells and the B cell developments, their differentiation and all these things. I have also introduced you a little bit to the T cell differentiation, that is, when a T cell, a naive T cell is activated. You remember the activation process, how a T cell gets activated by the different types of interactions with the dendritic cell primarily or with the macrophages, they get activated. So, with the APCs, with the antigen presenting cells, and one of the major cell types of this type of cell is the dendritic cell, because dendritic cell is kind of the cell that is connecting between the innate and the adaptive system. So, it is a cell of both innate system, adaptive system, although there are very classical differentiation between the cells of the innate and the adaptive system. So, when we call the cells of the adaptive system, we usually understand the B and the T cell. When we say cells of the innate system, we usually understand the neutrophils, basophils, mast cells, macrophages. These are the cells of the innate system. I already you got an idea in the previous classes, or I have already told you several times and discussed it, that the 2 systems of the immune system, the 2 systems, the innate and the adaptive system, they are very well coordinated. So, it is not that the innate system works discretely or the adaptive system works discretely. So, they are very well coordinated. There is a coordination between these 2 systems and they try to help each other. So, like the innate system, when it fails to kill a pathogen, when it fails to destroy a pathogen and clear an infection, it transmits that signal to the cells of the adaptive system. Once the cells of the adaptive system gets activated by the antigen presenting cells or these macrophages, dendritic cells, they get activated. Once they are activated, they again come back and help the cells of the innate systems to clear the pathogen. And these cells, which actually help in the infection clearing or clearing of the infection, that means which are involved in these immune responses directly or indirectly, they are known as the effector cells. So, they are the effector T cells. So, for example, T cells, if you go back to the activation and differentiation of the T cells, you will see that the T cells, be it a CD8 plus cell or a CD4 plus cell. So, CD8 plus cell will develop into a cytotoxic cell, CD4 plus cell will develop into a helper cell. Be it a CD8 or be it a CD4, you have either the memory cells or the effector cells. And the effector cells are those cells which ultimately helps in either they directly go and attack the pathogen and kill the pathogen, or they will be involved in indirectly, they will try to help the other cell types. They can also help the cells of the adaptive system. So, there are T helper cells that are responsible for helping the B cell development, for example, or B cell proliferation. If you have so I have discussed this in the last few lectures as well. So, you have seen how the T helper cells, they help in the B cell development, proliferation and activation. So, they are helpful cells, they help other cells in many ways. They also help the cells of the innate system, and that is why they are known as the effector cells. That means the effector T cells, they are the T cell effectors. Now, how these cells of adaptive system, they become effector T cells, that is a big thing. So, that has been, so there has been a lot of research, a lot of understandings of these mechanisms, and people try to understand how these cells, they actually become effector cells, which were the cells of the adaptive system, how they get activated and become the cells of the adaptive system. You have learned the activation part by now. So, if we quickly go back to our immunological overview that we have discussed earlier, we remember this, this picture where we have seen that T cell, a naive T cell, let us say this is a CD8 plus or a CD4 plus naive T cell, can interact with an antigen presenting cell like a dendritic cell by the presence of the MHC molecule on the APC, on the antigen presenting cell, which presents the antigen, with the TCR or the T cell receptor and the co-receptor, CD4 or CD8 co-receptor, and there are many other interactions we are not discussing here, whatever. So, once these interactions occur, then it is destined, then it is committed to become a T cytotoxic or a T helper cell. A T cytotoxic cell is also an effector cell, and we know, or we have learned in some of our earlier lectures, how the T cytotoxic cells can, they directly have killing effects. So, they are the direct killers. So, they can directly go and kill, they can inject enzymes, they can inject bactericidals or pathogen killing materials into the cells, into the infected cells, and that is how they kill. So, we have learned the effector mechanisms a little bit about the T cytotoxic cells. Now, what are the effectors or the effector cells of the T helper cell type? So, if we go and see what are the helper cell types, and we will see that the helper T cells, they can develop into different effector T cells as well as certain other T cells which are not really the effector cells, but they are involved in some other processes like the TFH or the T follicular helper cells. We know them very well because we have learnt about their role in the B cell development process, and the T reg or the T regulatory cells, and they are also very important in regulatory roles. So, they have regulator, they are the regulators of the T cell responses. So, these are the two different types which are not really involved in the effector mechanisms or the effector responses, And the three major effector T cells that are involved in the effector mechanisms, they are the Th1 or the T helper 1 type, the Th2 and the Th17. I already told you a little bit in my previous lectures probably how these cell types, they differentiate into different cell types. So, it is primarily controlled by the presence of the cytokines, the interleukins. So, depending on what kind of interleukin or what kind of cytokine is present in the surrounding, it will develop into a Th1 or a Th2 or a Th17 cell type. So, for example, if there is a lot of IL-12 in the surrounding interleukin 12, then it will develop into a Th1 cell type. If there is a lot of interleukin 4, IL-4, it will develop into a Th2 cell type, and if there is a lot of interleukin IL-6 and along with that, not only IL-6, along with that TGF beta together, then it will develop into a Th17 cell type 17. And the Th17 is primarily named as Th17 because it is one of the major sources of the interleukin IL-17. So, these are the three major cell types or the effector cell types that are being produced in presence of the different cytokines. We will discuss cytokines very soon in our upcoming lectures about the different types of cytokines, their mechanisms, how they work, what they do, all these things. So, for the time being, let us just learn the names of the cytokines and their actions and their how they help in these effector mechanisms involved in this helper that are induced by these helper cells. So, now this T helper type 1, type 2 type, this Th1, Th2 and the Th17, they can help in distinct mechanisms. So, what are the distinct mechanisms that the Th1 cell is involved in? So, the Th1 cell is primarily involved in clearing of intracellular pathogens. That means, what is intracellular pathogens or intracellular bacteria? Intracellular pathogens means those pathogens or bacteria that has been ingested or phagocytosed by a macrophage, for example. So, in the tissue, when there is a if you look into the tissue here, in the tissue when there is a tissue damage or any insect bite or whatever, then there is a lot of pathogens that is inrushing, that is coming in, and these pathogens first they meet the cells of the innate system, and they meet the neutrophils, the macrophages, the mast cells and all this. So, now these macrophages, they can engulf the bacteria. So, once these pathogens they enter inside the bacteria, the cells of the innate the cells of the adaptive system, for example, these Th1 cells, they can help in activating the macrophages to become cytotoxic. So, they themselves are not cytotoxic, but I mean these T helper cells themselves are not toxic. So, they are not cytotoxic, but they can release some cytokines, give some signaling molecules, so that they can turn these macrophages into cytotoxic cells, and they can activate and enhance the killing by the macrophages. So, these cells of the adaptive system, they come to the periphery or come to this, they get activated, then they differentiate, and then they move through the bloodstream, and then they come to this damaged tissue area. So, they infiltrate this damaged tissue area, and there they start to show their effector functions. How do these cells of the, or what signaling or what exactly directs these cells to this site of action? So, these cells, normally the naive T cells, they express certain types of cell adhesion molecules. I have already discussed about the cell adhesion molecules a little bit in my inflammatory response classes. If you remember from that classes, that CAM, we have discussed about different types of cell adhesion molecules like the selectins, the mucins, the immunoglobulin like CAMs, the ICAMs. So, these CAMs are expressed, or the cell adhesion molecules that are expressed on the surface of different cell types. They are expressed on the surface of the endothelium, they are expressed on the surface of the lymphocytes, many other immune cell types. So, these CAMs, or the cell adhesion molecules, they help in adherence and migration of the lymphocytes from one place to the other. So, they are basically the helpers in migration, and these cell types, these matured, finally differentiated matured cell types, developed T cells, effector T cells, they can express different types of CAMs on their surfaces, as well as they can also express different types of chemokine receptors. We will discuss what chemokines are. For the time being, let us understand from the name that chemokines are the chemoattractants. That means they attract some cell types. So, chemokines are small molecules which can attract a cell type by binding to the specific receptors present on the surface of the cell. So, for example, a chemokine can bind to a specific chemokine receptor, and so it is a specific receptor ligand interaction. So, the chemokine is the ligand which binds to its specific receptor and attracts or pulls that cells. So, it is like a magnet to iron interaction that is occurring, and this interaction is very specific. So, it can draw the cells or pull the cells to a specific location. So, that is how these cells of the adaptive system, these T cells, these fully differentiated and developed T cells, they now start to express different types of chemokine receptors. For example, a naive T cell usually expresses on its surface a CCR7. As soon as this naive T cell starts to differentiate into different types of T cells, effector T cells, their expression of CCR7 goes down, and then they start to express different other chemokine receptors, and different T helper cells, they express different chemokine receptors. For example, these cells, these Th1 cells, they express CCR5, the Th2 they express CCR4, and the Th17 they express CCR6. So, they start to express that like the naive T cells, they were expressing CCR7, and once this differentiates into the different T helper cell types like the effector cell types, they start to express different chemokine receptors like CCR5, CCR4, CCR6 on their surface, and by virtue of that, because of that, they now start to move to different locations and start to show different responses, different immunological responses. So, what we were discussing about is that these cells, the Th1 cells, they are primarily responsible for intracellular pathogens. They are responsible for intracellular pathogens. Then we have the T helper cell type 2, which are mostly responsible for parasites, for killing of parasites and helminths. Because they basically go to the they changes the mucosal smooth muscle motility, and when there is a parasitic infection or a helminthic infection, particularly in the stomach or in the intestine, so they changes the mucosal smooth muscle motility and tries to clear that helminthic attack or the parasitic attack, how we will discuss. So, these are the Th2 type cell, and the Th17, they actually elicit a class of responses which are responsible for extracellular pathogens. So, like the Th1 is responsible for intracellular pathogens, these are responsible for extracellular. So, those which are not being internalized, that has to be dealt with. They are mainly dealt with the Th17 class of cell types, the effector cell types. Now, what these Th1 cell types they do primarily, one of the major functions is that they activate a macrophage. So, they can activate the macrophage. And how do they do that? So, these are for example, a Th helper type 1 cell, and this is a macrophage which has engulfed a pathogen. So, with a pathogen inside, that means intracellular pathogen in here, and that can present, that expresses the MHC on the surface and that interacts with this T cell by the T cell receptor and the CD4 co-receptor, and as well as there is a, because this matured, this differentiated matured Th1 cells, they express on their surface the CD40 ligand, CD40L. We by now we have known about the CD40 and CD40L interaction from our B cell development processes. So, they express on their surface the CD40 ligand, and these macrophages they express the CD40. So, now they can now interact with the CD40 and CD40 ligand interaction occurs, and after this interaction occurs, there are, of course, there is signal transduction, more gene transcription, downstream transcription of more genes, and that leads to the production of different cytotoxic materials, some cytokines that will go and kill the bacteria more efficiently. So, this is one of the functions. So, they now kill the bacteria or the pathogen, primarily the bacteria, they kill the bacteria or the pathogen more efficiently. And one of the major signaling in this entire process is that these T cells, they starts to release a specific cytokine which is known as interferon gamma. So, INF gamma, we will learn about these interferons, we will learn about all these interferons and the cytokines in our later classes very soon. So, this they start to express, they start to produce this interferon gamma, which has a very important role in activating these macrophages. So, now these macrophages they become activated. A second mechanism by which these Th1 cells they work is also they increases the production of the monocytes, monocyte production in the bone marrow, and that is primarily done due to cytokines like IL-3. Then they also produce a very important cytokine that is known as TNF alpha. We will learn about TNF alpha and its roles primarily in later in our cytokine classes, but this TNF alpha is one of the very important signaling molecules. They can activate the endothelium, they can increase the vascular permeability, and they allow more macrophages to come into the blood. They can increase vascular permeability and allows more macrophage entry into blood. Now, once more and more macrophages comes out in the blood, that has to go to the site of action as well. So, now macrophages which are coming to the blood, they has to now migrate to the site of action and show their action. So, they have to do their action, do their function, and for that you need some chemokines, for example. So, one of such chemokines is CCL2, that is also released by this Th1 cell type, and they can attract more macrophages. So, CCL2 is one of those chemokines that attracts more macrophages. They can attract more macrophages to the site of action. Or the site of infection. Okay. So, the Th1 cells at least performs 4 different effector functions that we can summarize here. That is, they can produce interferon gamma, they can activate the macrophages and induce cell killing, bacterial cell killing more and more. They can also induce production of the monocytes. They can also allow more macrophage entry into the blood, and they can also attract those macrophages to the site of action. Coming to the action of the Th2 cell types. What are the Th2 cell types? So as I told, the primary action of the Th2 cell types is to increase the motility of the mucosal smooth muscle. So they activate the mucosal smooth muscle, they activates the mucosal smooth muscle, increases the motility. So increases motility, and by that they helps in clearing of the pathogen, primarily the helminths, the worms that infects our intestine, for example. So those are dealt with this kind of Th2 responses, type 2 responses, and they can also activate the eosinophils and the basophils, for example. So they activates the eosinophils, for example, and activation of the eosinophils and the basophils would lead to the release of many cytotoxic molecules. So they release a lot of cytotoxic material or molecules that can clear the pathogens, primarily the parasites. So they deal with the parasites, as I told, and they also secretes a class of interleukins like IL-3 or IL-9, for example, and they can activate the mucosal mast cells. So they activates the mucosal mast cells. The mast cells, this is another mechanism. So this is another mechanism of amplification of the signal. They can activate the mucosal mast cells, and that mast cells, they are granulocytes. So they will start releasing the granules like the histamines, for example, the histamines, the prostaglandins, and that will basically this release of this histamines or these prostaglandins, so these vasoactive amines, that would lead to also increase the fluid flow in the mucosal lumen. So a basic mechanism by which this Th2 response works is sometimes also known as a whip and sweep mechanism. So it's also called a whip and sweep mechanism, whip and sweep mechanism. So they sweep out the pathogen from the intestine or from the body and try to clear that pathogen, primarily the helminths and the parasites. And a third effector mechanism is the Th17 response, and the Th17 response also it induces the epithelial cells. So they induce the epithelial cells and tries to, so as I told that these are the Th17 response primarily deals with extracellular pathogens. So the pathogens which has not been ingested or which has not been internalized. So that means they help certain cell types for release of cytotoxic material or release of some bacteriostatic or bactericidal material. So they primarily induces the, so they produce interleukin 17, as I told that they are the primary producers of interleukin 17, which activates, they activate the epithelial cells, and that leads to the production of the antimicrobial proteins. So produces antimicrobial proteins and bacteriostatic and bactericidal antimicrobial proteins, peptides, and leading to bacteriostatic or bactericidal effect. This also, they also produce, they are the primary secretors of IL-17, as I told. So IL-17 also recruits more neutrophils to the site of action, neutrophils which are the fastest acting cell of the immune system. So, they go and infiltrate into the site of action or the site of infection. So, IL-17 is one of those cytokines that can actually bring more neutrophils to the site of action, and as well as they are also they secrete this CCL20, the CCL20, which is a chemokine, and the CCL20 attracts more Th17, sorry. So, they attract more of this T helper 17 cells to the site of action. So, quickly a quick look into this entire picture is that these effector cell types, the helper cell types, they are the effector cell types. There are the 3 different effector cell types: the Th1, the Th2, and of course the Th17 are the 3 major effector cell types. Th1 primarily is involved in intracellular, dealing with the intracellular pathogens, that means the pathogens which has been internalized. They help or activates the macrophages, they help in monocyte production, they allow more macrophage entry into the blood, and also migration of those macrocytes or of those macrophages into attraction of those macrophages into the site of infection. A second type of response is initiated by the Th2 cell types. They produce one of the major producer of IL-13, for example, interleukin 13, and which activates the mucosal smooth muscle, increases its motility, and primarily by the production of the mucus by the goblet cells. So, they can activate the goblet cells and enhance the mucus production, and by that they can also increase the motility of the mucosal smooth muscles. They can activate the eosinophils, then by that and recruit more eosinophils, and which can produce a lot of cytotoxic molecules that can clean up or clear the parasites or the helminths. And this is primarily done by the action of interleukin 5, which is also produced from the Th2 cells, and then they also produce IL-3 and IL-9, which primarily activates the mucosal mast cells, and mast cells, as you know, are the major sources of the histamine. So, mucosal mast cells are a small category of the mast cells that are present here, and they are activated by these Th2 cell types, and they can produce histamine or prostaglandin, leukotrienes, all these molecules, which actually helps in the vasodilation, and thereby increases the fluid flow to the lumen. So, to the mucosal lumen, and then by that action they can actually sweep out the pathogen. And thirdly, the Th17 cell types, they are mostly dealing with extracellular infection or pathogens, extracellular pathogens. So, they activates the epithelial cell to produce a lot of antimicrobial proteins. They induces the epithelial cell to produce a lot of antimicrobial proteins. They also produce, they are the major producer of the IL-17, interleukin 17. So, which actually attracts more neutrophils to the site of action, and they also attract more of the Th17 cell type to the site of action. So, we kind of today tried to summarize or get an overview of the different effector cell types. I try to give a very preliminary idea about the 3 different effector mechanisms, what they do, what the major functions are. You can get more details in the books, but we really do not have scope or time to go into more details. So, I will stop it here. For the effector T cells, primarily the T effector cells, we will stop the discussion here, and we will try to learn more about the different effector molecules, the effector mechanisms. We will try to learn about the complement proteins, the cytokines more in details, and then we will see how they work in the different contexts. So, what are the major roles of the different cytokines in different contexts in the innate and the adaptive system, what are their roles. So, we will discuss all those things slowly in our upcoming lectures in more details. So, that is all for this lecture, and thank you.